All right, guys, so here we are. At this point, we have talked through all the stages of mitosis. We've gone through meiosis. So this last chunk of your notes is just to sort of review a little bit about the differences between mitosis and meiosis, and then to talk about what happens when these cell cycles don't go quite the way your cells intend them to. Okay, so let's start off and talk about big picture stuff here. So we're talking about cell division, going from one cell, splitting that cell into two, or in the case of meiosis, going through a second round of division and getting 4 daughter cells at the end. So if we compare sort of major big picture things, mitosis, you start with a parent cell. That parent cell is diploid, all right, so it's a 2N cell. For humans, that means there's 46 chromosomes in that parent cell. We're going to go through one round of division, so your prophase, your metaphase, your anaphase, your telophase, and then finally cytokinesis to create 2 daughter cells that are also diploid. They're also genetically identical to that parent cell. So again, if we take human numbers, we're talking about going from a 2N 46 chromosome diploid parent cell and creating 2 daughter cells that are also both diploid cells, 46 chromosomes in each cell. Typically, we're talking mitosis going and creating body cells to create new body cells for things like repair and for growth. So when we talk about body cells, remember somatic cells are the more scientific term for that. So you end up with identical copies, all right, or that word clone in terms of the fact that we're talking about DNA from the parent cell being identical to the DNA in the daughter cells. All right. Asexual reproduction also falls into that when we're talking about things like your yeast creating buds. Remember, it's kind of growing off of its side, it's going to pop off, and it's now a mature individual to grow and continue to grow. Your fragmentation, like we saw in your sea stars. All right, or the sort of modified version of mitosis in bacteria, which we talked about as binary fission, because remember, they don't have a nucleus, so they can't go through true mitosis. All right, so that takes care of this side. Meiosis is really there for sexual reproduction. We're taking a parent diploid cell, so 46 chromosomes in humans, and making 4 haploid daughter cells. Remember, haploid would be N, so in humans, if we're taking a diploid 46 chromosome, we're making 4 haploid cells that would have 23 chromosomes each. All right. How do we do that? Well, remember, we've got to go through those 2 phases of division. Go back to your notes if you don't remember what that looks like. Okay, but we're going to go through meiosis 1, still prophase 1, metaphase 1, anaphase 1, so on and so forth. All right. By the time we get through that first round of division, we're already at haploid cells because we've taken that tetrad lining up in metaphase this time and splitting the homologous chromosomes. Those homologous chromosomes then are split into that first round of division, but the sister chromatids stay together. We go through the second round of division in meiosis 2, same process in terms of prophase 2, metaphase 2, so on and so forth. All right. But it's those individual chromosomes that line up in metaphase 2 and the sister chromatids then split in anaphase 2 to get us those 4 sex cells at the end. All right, remember that if it's a male, he's going to go from 1 germ parent cell, diploid cell, to 4 sperm at the end. Females have to give a little more in terms of cytoplasm and your organelles and all that kind of stuff, so it's going to be 1 parent germ cell to 1 egg cell. She'll make those other polar bodies, but they're too small to be useful, I should say, and so her body will reabsorb those. It's only that single egg at the end that's actually useful. All right. Meiosis also gets us not only haploid cells, but those haploid cells are genetically different from the parent. All right, remember we talked about genetic recombination. So that different DNA sequence, those different DNA traits all right, that are getting passed on to those offspring are different because of the independent assortment. So the way that those homologous chromosomes line up in metaphase 1 is completely random. Okay, so one cell could get 50% mom, the other could get 50% dad, but more than likely you're getting some variation within that. So 25% of mom goes one way, 75% of mom goes the other. All right, and then dad would make up the rest. You also have crossing over, which happens in prophase 1. All right, so taking that homologous chromosome and also mixing up information from what, say, your mom gave to you, what your grandparents gave to her. All right, so taking bits and pieces of the chromosome and mixing those up as well. All right. So here, if you take a look, this sort of lays out, so mitosis is all on this side. So here's your parent cell, prophase, you've got your duplicated chromosomes sitting here as sister chromatids. Okay, we go through metaphase, and those sister chromatids stay together, they're lined up, but we're not worried about whether these chromosomes are homologous to each other. Okay, we go through anaphase, telophase, and cytokinesis, which is what they're showing you all here at one shot. And your sister chromatids have split, your cytoplasm has split. All right, and so you've got two 2N cells, so two diploid daughter cells. All right, and here's the original parent cell, which was also 2N. Okay, so we're staying diploid to diploid. This side over here, we're talking meiosis now. So they're showing you some crossing over, so you'll take a look at your homologous chromosomes here. All right, one from mom, one from dad, and we're taking that information and kind of swapping some of that. All right, and synapse also happens here to get those homologous chromosomes together. Here we want to keep that information the same and keep it together. All right, so that when we get to metaphase 1, it's the homologous chromosomes, those tetrads that line up, not just a single chromosome. All right, we go through anaphase 1, telophase 1, cytokinesis 1, and it's going to be those tetrads that split. So one half goes one way, the other half goes the other, but those sister chromatids stay together. All right, by the time we go from here, here's my original parent cell, duplication of my DNA. All right, so my 2N here. I go through my first round of division, and I'm already at a haploid cell. Okay, so I've taken the information and split it. Remember I said it's like taking a textbook. We've ripped the textbook in half by the time we get here, but you still have a photocopy of part 1 of the book and a photocopy of part 2. That's why your sister chromatids are still together. We go through meiosis 2, all right, and metaphase 1, your chromatids will line up, and now you end up with your sex cells here at the end. All right, so you still have those N cells because we're already haploid here. All right, if this is a male, you get 4 sperm. If you're a female, you'll get one of these as a viable egg. The others will be polar bodies that her body will just reabsorb and get rid of because they don't have all of the other stuff, the mitochondria, the cytoplasm, all that kind of stuff that new baby is going to need. Okay, so mom's kind of doing a 1-to-1, dad's going to do a 1-to-4 split. All right. Okay, so